Welcome to this explainer. Today, we're decoding the builders. It's a cellular detective story all about proteomics. You know, when we talk about biology and the secrets of life, almost everyone instantly thinks of DNA, right? It gets all the glory, the magazine covers, and those famous double helix statues. But what if I told you there's a much, much bigger mystery hiding inside our cells? A sprawling, dynamic world that goes way beyond just our genetic code. Today, we're cracking open the fascinating, real-time world of proteomics to see what actually builds our bodies. Let's get right into it. To really grasp why this matters, we first need to look at the fundamental difference between DNA and proteins. Think of DNA and its messenger, RNA, kind of like the architectural blueprint for building. I mean, it holds the instructions, sure, but a piece of paper has literally never built a house on its own. Proteins, on the other hand, well, they're the builders. Our source material actually puts it perfectly. It says proteins are the main effectors of all cellular functions. Basically, they're the ones causing the effects. They are the construction workers, the foremen, and the heavy machinery making absolutely everything in your body happen. Breathing, moving, thinking, all of that is proteins hard at work. But here's the catch. Just reading that genetic blueprint, it's not enough to understand the building. Why? Because proteins have this incredibly complex life cycle that you just can't study using DNA or RNA techniques alone. Imagine a protein as a factory worker. Just knowing their name on an employee roster doesn't tell you what they're actually doing on the floor today. Once a protein is made, it goes through maturation and processing, it gets sorted, sent to specific areas, and it can undergo what scientists call PTMs. That stands for post-translational modifications. If translation is the moment the worker is hired and created, these modifications are everything that happens after. It's like our factory worker putting on a specialized hazmat suit or grabbing a specific power tool to do a totally different job. Then they team up with other workers, those are protein-protein interactions, before eventually retiring through a process called degradation. Because of all this constant activity, just measuring RNA doesn't tell you what the proteins are actually doing. And this brings us to the first massive challenge in the field of proteomics. The sheer complexity. See, our human genetic blueprint only contains roughly 35,000 genes, and of those, maybe 15,000 are expressed in a single cell at any given time. But here is the kicker. Because of all those modifications, the outfit changes, the tool swapping we just talked about, those 15,000 base expressions can result in anywhere from 50,000 to over 150,000 chemically different protein species. So one single gene can actually produce dozens of different variations of a working protein. It's a massive multiplier. All of this complexity builds into an even bigger puzzle. If you look at the scales and the hourglass here, they brilliantly illustrate two massive hurdles: dynamic range and plasticity. The scales represent abundance. Inside a cell, the difference in number between the most common proteins and the absolute rarest ones is huge, like a factor of 10 to the fifth power, or even 10 to the sixth power. To put that into perspective, trying to find a low abundance protein is like trying to hear someone whisper to you while you're standing in the front row of a heavy metal concert. The loud stuff just completely drowns out the quiet stuff. And the hourglass? That represents plasticity and turnover. The protein landscape inside us isn't some static photograph. It is a continuous, rolling movie. Proteins are constantly changing their expression patterns, getting modified and breaking down. So, the million-dollar question is, with all this mind-boggling complexity, a massive dynamic range, and this constant shifting, how on earth do we actually study them? Well, we hit another wall, and it's summarized perfectly by this brutal fact from our source. Proteins cannot be amplified. Think about it. If you want to study a tiny, rare piece of DNA, scientists have a neat trick. They essentially act like a cellular photocopier and multiply that DNA millions of times in the lab until they have enough to study. But you absolutely cannot do that with proteins. With proteins, what you have in your sample is exactly what you get. Going back to that heavy metal concert analogy, you can't clone the person whispering. You just have to figure out how to build an incredibly sensitive microphone. So how do scientists play cellular detective and actually solve this? They use a highly specialized two-step investigative workflow. Step 1 is to separate, and step 2 is to analyze. We absolutely have to separate them first because proteins have vastly different physical and chemical properties. Some are acidic, some are basic, some love water, meaning they're hydrophilic, and some completely hate water, so they're hydrophobic. To handle this, we use techniques like electrophoresis or liquid chromatography. Now, those sound like big words, but they essentially just act like high-tech molecular sieves or obstacle courses. They force these vastly different proteins to line up so we can actually look at them clearly. This really beautifully illustrates exactly what that separation step looks like. Notice that jumbled cluster of colored circles on the left side? That is our raw, chaotic protein sample straight from the cell. But push that messy cluster through our separation techniques, sorting them out based on those chemical properties we just talked about, and what's the result? You get that neat, orderly line on the right. It's kind of like a highly efficient bouncer at a busy club. The separation step ensures that all the suspects walk through the door one by one, perfectly prepped and ready to be fed into the ultimate analytical machine, the mass spectrometer. And once they enter that mass spectrometer, we get a result that looks exactly like this graph. Now, if you're new to this, it probably just looks like a bunch of tall red spikes. But what you're actually looking at is the chemical fingerprint of a protein. See, the mass spectrometer essentially weighs the protein fragments with just mind-boggling precision. The tallest spike you see here represents what we call the monoisotopic mass, which is really just a fancy way of saying the exact weight of the most standard version of that protein. And that cluster of smaller spikes right next to it, that is the isotopic envelope. Because atoms out in nature sometimes come in slightly heavier variations, the machine is so incredibly sensitive that it actually picks up those tiny, tiny weight differences too. This creates a highly precise, distinct barcode for every single protein species. All right, so we've got our chemical barcode. What is the final step? We match it. Just like a detective running a fingerprint through an international criminal database, scientists take these mass readings and bounce them against massive global protein sequence databases. And matching these precise barcodes reveals exactly who was in the cell. For instance, we might find a match for DHX9_human, which is an RNA helicase, basically a worker that unwinds genetics. Or maybe MIO6_human, also known as myosin V, which is a worker that transports cargo around the cell. We even see matches across species, like TP2A pig. By doing this, we can finally name the exact builders, wearing their exact outfits, doing the specific work inside the cell at that exact moment in time. It's honestly amazing. Which leaves us with this final, truly thrilling thought. We are no longer just looking at the DNA blueprint and guessing what the building looks like. By separating the chaos and weighing the chemical barcodes of these tiny cellular workers, we can literally see the exact mechanics of life in real time. So I'll leave you with this question to ponder. Now that we can accurately fingerprint the dynamic, ever-changing builders of our cells, what previously incurable diseases, ones driven by malfunctioning proteins, might we finally be able to understand and actually cure? The frontier of proteomics is wide open, and it is a total game changer. Keep exploring, and thanks for joining me for this explainer.